Our next speaker really doesn't need an introduction, but other than his name, we have Dr. Stephen Treon, who's going to be talking about treatments on the horizon. And for those of you that are new to the Ed Forum or are new to WM, he's been a luminary for all of us for decades. I'm kind of dating myself also, and probably him. And really, you know, and just really briefly, I just remember he hung his hat on Waldenstrom's patients and Waldenstrom's research, and has spearheaded the only organization or the only institution that focuses specifically on Waldenstrom's patients, to the point that it's not just him there, there's also others that are focusing on Waldenstrom's patients. That's a testament to his vision, a testament to his fortitude, a testament to his capabilities, and he's well regarded not only in the Waldenstrom's medical community, but in the global medical community in general. Please give a round warm of applause to our next speaker, who's been talking about treatments on the horizon. If anyone knows what's coming, it's Dr. Treon.
And thank you so much for that very kind introduction. And I want to just also ask for another round of applause for Jeff's excellent talk. Really appreciated it. It sets really a nice introduction for what I'm about to talk to you about, but I also want to acknowledge that Jeff, as well as our colleague Christian Buskey and I had the opportunity to organize the 12th International Workshop, which took place in Prague. And it was an outstanding meeting because of the efforts of Chris Patterson, who served as our secretariat. And if I can ask you for a round of applause, because the international workshops on Waldenstrom really are how our scientific community is able to connect. And in fact, I'll show you in just a moment a slide that shows you a picture from that meeting, but we had over 400 investigators who came to Prague for the 12th International Workshop. Contrast that to the first international workshop, where there were only 19 of us. And in addition to 400 live people in Prague, we also had over 200 that joined us virtually. So it does show you how the global family has truly grown around Waldenstrom's. I'll show you pictures of folks as we make our way through the slides today, because I really want you to get to know these people and just know how hard they're working on our behalf to help us advance, getting to the cure of this disease. So these are my disclosures, just to put those out to you. And this is actually the 12th International Workshop that was held in Prague. Our intent had been to hold this meeting actually in Jerusalem in Israel, but unfortunately, because of the events of October 7th, the meeting was relocated to Prague. But it was really a successful meeting. We ended up having quite a bit of knowledge that was exchanged. Importantly, we also had 6 consensus panels that assembled. I'll talk about one of the consensus panels later in my talk, but these consensus panels were made up of world experts focused on Waldenstrom's that tried to address different elements of care for our patients. Now, I think right off the bat, it is important to acknowledge this is an interesting disease because patients can present very differently. And it sort of sets the stage that when we're thinking about treating Waldenstrom's, we have to also keep in mind what we're treating and what the symptom is that is bringing a patient to need care. And for many of the patients, it's anemia or their blood counts are down because of the infiltration of disease in the bone marrow. But I want you to also come away understanding that the IgM that's produced by these cells can also wreak havoc in many different ways. Also, there are patients who get the disease in their lymph nodes and spleen, so you have to start thinking more like a lymphoma doctor if that's the case. And in some more rare circumstances, we do see patients with Bing-Neill syndrome. This is when the disease involves the central nervous system. But again, all the consensus panels that I mentioned to you tried to deal with different elements of Waldenstrom disease. Now, traditionally, we have cared for this disease with chemotherapy using drugs like proteasome inhibitors like bortezomib and bendamustine, cyclophosphamide, which many of you are aware of. combinations with rituximab, a drug that I had the pleasure of helping to develop some 30 years ago. With these combinations, we've gotten very high response rates, as you can see, but what we still lack is complete remissions, because it's very unusual to actually be able to get a complete response with our current therapy. And as Paul Kitchen is challenging us these days to think about a cure, these are the numbers we need to move upward. We need to see more complete remissions before we even start thinking about curing Waldenstrom's, which is why I want to set the stage for the horizon of why we need to look beyond the horizon, because these are the numbers that we hope to improve on with our clinical trials and opportunities that we now have. Also, to keep in mind that many of these combination therapies that we used to use also had side effects. I don't want to dwell on this slide too much, except to say that when we saw the side effects, both short-term and long-term, of these chemoimmunotherapy approaches, we recognized why we had to do better, because we had to come up with better targeted therapies that were more effective for our disease. And this was really the problem, and I think in many ways continues to be the problem around Waldenstrom's, and that is that in the old days, we had to wait around until some of these drugs were vetted out in other diseases, lymphoma, myeloma, CLL, and then at some point, they'd be handed down to us. So a lot of the medicine that we practiced was what we called hand-me-down medicines. So this had to change, and this is why I want to now celebrate with you these opportunities that we now have based on the advances that have been made. And I want to thank two entities for helping us open up these new opportunities. One is this individual, Peter Bing, which you probably haven't heard too much about. But Peter Bing was the chairman of Stanford University and really one of the great philanthropists in this country. And Peter, along with the International Waldenstrom's Macroglobulinemia Foundation, and I show you the old logo on here just to remind many of you of the older days with the IWMF, came together to fund a very important project that allowed us to do whole genome sequencing of 30 Waldenstrom patients in search of what the cause of the disease was. Up to this moment in time, we didn't know what caused Waldenstrom's. There was no really genetic element that we could point to and say that's the reason why this disease exists. So in 2011, we made this big breakthrough, thanks to the funding by the IWMF and Peter Bing, and we discovered the MYD88 mutation. This appeared in the New England Journal of Medicine, and I got to say, if you're thinking about career highs, whenever you get something in the New England Journal of Medicine, it's important and exciting. And we noted in that study that 90% of Waldenstrom patients actually had the MYD88 mutation. And this, that year, was the best of the American Society of Hematology. This is our big meeting in the hematology sphere. It was the best of ASH for that year, meaning that it was picked as the top discovery for that year, and it appeared in the New England Journal the next year. Now, Zach Hunter, along with, and you met Zach yesterday, and Leanne Zhu in our group, then went about making the first assays that could actually detect this mutation. And this was published in very prominent journals as well, but it also actually ended up doing something very interesting, and that is that it showed us that with this more sensitive technology, the MYD88 mutation could now be found in about 97% of all Waldenstrom patients. And almost all the patients had this recurring mutation called the L265P mutation, which means, and those of you who may remember your basic biochemistry, L is leucine, the amino acid, P is proline. So what that means is that at position 265 on MYD88, leucine got substituted because of a mutation for proline. And proline is a troublemaker. It's a amino acid that's very rigid, and it turns around the structure of the protein in a way that it now allows it to signal independent of some of the growth controls that exist. And so MYD88 can now fire and continue to cause havoc. And I'll show you exactly what it does. And it was their prowess that now opened the door to the molecular testing that many of you perhaps have seen, through your laboratories in trying to discover this mutation. Now, another very hardworking individual in our group spent the next decade after this discovery trying to tell us exactly what MID88 does. And this was Guang Yang. And I really think we need to celebrate him as a tremendous hero to our cause because he was able to make some interesting observations. The first was that MID88 is part of a signaling network that's there as an alarm system in case we see an infection, virus, bacteria come into contact with our immune cells. And this is part of that firing mechanism. And so when you're in the normal state, you need to see the virus or bacterium or one of the products come in contact with the immune cell to fire off MID88. But when you have the mutation, it's firing off on its own. It doesn't need an external signal anymore. So it's almost like the light switch got turned on and then it got broken in that open position. And that's what's happening. And then what Guang was able to figure out is that there were a couple of things that were occurring because of this switch being left in the open position. The first was that you see this molecule HCK, it's dead center there in blue. This molecule actually doesn't belong in a cell like Waldenstrom's. It's usually downregulated and it goes into oblivion, but because of the MID88 mutation, it's actually upregulated, it's turned on, and then see all the cascades that come off of that, meaning that HCK now is turning on all these other light switches that are allowing the cell to grow and survive. Among them, you can see BTK, that's shown there in red. And this is when we understood the opportunity to drug the MID88 pathway by developing BTK inhibitors. And of course, the first that we developed along this pathway was ibrutinib. Now, I want to tell you why I'm really excited to share this with you, because ibrutinib not only became the first drug ever approved specifically for Waldenstrom's, but because we were able to merge basic science, genetic science, translational science, along with clinical results that we were getting, this was also the drug that also got the first breakthrough indication for any oncology indication. So when you go to the FDA, there is this special pathway that allows you to get accelerated approval, but it's when you can come with good science data married with some good early clinical data that earns you this. And the fact that this was actually earned in Waldenstrom's, we should all be excited about, because it opened the pathway for all oncology indications, regardless of the drug, by showing that this pathway was tenable. And of course, then came the FDA meeting where we could present the initial results from the ibrutinib trial in Waldenstrom patients. These were all patients that were previously treated. And in June of 2014, you can see Leah Palumba there right next to me, and myself went to the agency along with many other stakeholders from Janssen and Pharmacyclics, and we had a very favorable review leading to the approval of ibrutinib for the treatment of Waldenstrom's. And of course, what I then want to tell you is when you open the door for an approval, all of a sudden you get a lot of excitement by other companies that also want to develop their BTK inhibitors in this space. And as a result, look at all the trials that have occurred that followed our trial that then opened the door for other drugs, including zanubrutinib, which is now also FDA approved for the treatment of Waldenstrom's. But you can see other drugs up there too. There's acalabrutinib, there's tirabrutinib, which is approved in Japan. And now we also have pirtobrutinib, which I'm sure you've heard a little bit about, but I'll tell you a little bit more about it today. And so I'm going to just summarize all these trials by telling you that when you look at BTK inhibitors, they represent the single in-class best drug that we have for the treatment of Waldenstrom's as a single agent. And in fact, over 90% of patients get a response. Those deep responses, which we call major responses, occur in about 80% of patients. And VGPR means that we can get 90% reduction in disease burden, and that occurs in about 30% of patients. The more important thing to keep in mind is that we can also keep the disease under control for long periods of time. And 3/4 of the patients on these different trials remain without their disease progressing at 4 years' time. These are really outrageous numbers. But in this, you should also see that there is something that's missing, and that's complete remissions, complete responses, because in aggregate, that complete response rate is 0. We don't get people in complete responses with these drugs, which is why we need to continue to do better. Now, one of the reasons why we're held back in being able to attain those deeper remissions is because of this mutation CXCR4. And I think many of you heard about this from Dr. Hunter's talk yesterday. But because of Dr. Hunter, we were able to discover the CXCR4 mutation. He went right back into that database that we had that initially allowed us to understand that MYD88 was our key culprit. And he was able to mine that data, that whole genome sequencing data, and discover these CXCR4 mutations. These actually, do occur very rarely in other disease settings, but they're almost exclusive to Waldenstrom's. Unlike MYD88, you can find MYD88 in other lymphomas too, but CXCR4 is fairly exclusive to Waldenstrom's. And so in our lab, we spent a lot of time trying to understand exactly what these mutations do. So did Irene Gobriel in her lab. And this is what we learned, that about 40% of all Waldenstrom patients have this mutation. And as a result of the mutation, you see a couple of things. One, the disease presentation can be altered. People especially that have one of these mutation types called the S338X mutation are the ones that appear with hyperviscosity syndrome with the very high IgM levels. The other is that this mutation also contributes to drug resistance. And I want to show you the next slide here because it's explained that in all those trials that I showed you with BTK inhibitors, there was a lot of work done around understanding what CXCR4 does. And let me just tell you, if you have the CXCR4 mutation, it'll take longer to get to one of those, deep responses. Your response won't be as good as somebody without it. And the time that the disease is kept in check is much shorter with CXCR4 mutations, which is why we've looked at this also as a potential target. I won't be talking very much about that today, other than the fact that some of the BTK inhibitors can help us overcome this, but I do think this is one of those mutations that we need to keep on our radar screen to develop specific drugs. And maybe during our Q&A, I can help address what our efforts have been. Now, when you have all these BTK inhibitors, the big question is which one and for which patient. And I think this is actually a really important topic before we set the stage for the next horizon. As many of you know, there was this very large study called the ASPEN trial where ibrutinib was compared to zanubrutinib. And this trial actually is what enabled zanubrutinib to get approved. And there were two key figures here, Konstantin Tam, as well as my other colleague and friend Thanos Dimopoulos, that helped spearhead or oversee this trial. It was made up of two groups. I think Jeff showed you very nicely that there were two groups here. The first one was patients who had the MYD88 mutation. The second was the patients that didn't. And if you want to learn one term today, these patients are called wild type, meaning they have no mutation. So if you hear me talking about MYD88 wild type, it's not that MYD88 is wild, it just means that it's the normal unmutated type. So cohort 1 was MYD88 mutated, cohort 2 was MYD88 wild type. What did we learn? We learned actually that both drugs are pretty good. The response rate, especially the deep remission response rate for MYD88 mutated patients was about 80%, regardless of which arm, maybe slightly more VGPRs with zanubrutinib. But we also saw that those patients that didn't have the MYD88 mutation responded very nicely to zanubrutinib. We didn't see that with ibrutinib when we did our original trial. And so this opened up now the opportunity to use zanubrutinib in this patient population. So what did we learn from this trial that really got us all fired up? And that is that those patients that were on zanubrutinib actually did better if they had the CXCR4 mutation. And so CXCR4 mutated patients seem to do better with zanubrutinib, not only getting into more deeper responses, but also those progression-free survival curves that Jeff showed you earlier. You can see that, at 42 months, 73% of the patients on zanubrutinib did not progress, whereas, as I can see here, 49% of the ibrutinib patients still remained without progression. And so this does show you a very large difference, and it's why we prefer zanubrutinib in the CXCR4 mutated population. Of course, as you've already heard from others, there was also an advantage in terms of side effects. With ibrutinib, there was more atrial fibrillation and more hypertension. With zanubrutinib, there was more neutropenia, meaning the white blood count was lower, but this did not result in any infection. So overall, the safety profile for zanubrutinib appeared very favorable as a result of this trial. And this is what the FDA felt was a means for approving zanubrutinib. I was lucky to be actually at both approval meetings as well as the zanubrutinib meeting. And so it was important for us to advocate the need to bring on other BTK inhibitors. Now, very big question that exists now in our field is, okay, we've got BTK inhibitors, we can get all excited, but we also have other drugs, and you've heard about bendamustine in this setting. And I think, this is still a very active debate in our field as to what should we offer patients initially. Should we do BTK inhibitors or should we do chemoimmunotherapy? Let me show you what we learned from the 12th International Workshop, which I think is very important. And I think in large part we should give Jithma Abakun from the Mayo Clinic in Rochester really a loud, round of applause, because very young, very motivated. He took on this multinational study looking at, patients who got bendamustine Rituxan, compared it to those that got ibrutinib. These were all patients that had not seen previous therapy but required therapy. And it was truly a multinational study made up of centers, in North America as well as Europe. And what we learned from this study was very interesting. And it's a retrospective study, and I think it's always important to keep that in mind. You do collect data and you see how patients did and you report it out. But what Jithma actually found was that patients who got bendamustine and Rituxan actually got deeper responses than those who got ibrutinib. However, and this is really the big however, it made no difference in terms of progression-free survival and overall survival. And he updated the data with 6 years now follow-up to show us, in fact, the numbers are the same. So you don't go wrong being on one therapy versus the other in terms of efficacy. Now, at the 12th International Workshop, we also learned from the Philo group. This is the French Innovative Leukemia Organization, and I put the French flag up there to remind us. And my good friend and colleague, Veronique Leblond, who decided after decades of service to Waldenstrom to retire in Prague at that meeting, had this final presentation to make. And that was a study with bendamustine Rituxan in Waldenstrom's. And she provided the long-term results of how patients did with bendamustine Rituxan who were treatment naive and required therapy. Now, this was a very exciting study because we've been looking at this as sort of guidance for a number of different reasons. One is to see the efficacy. You can see, in fact, major response rate, 96%, even those deep VGPRs, 56%. And when you look at the complete remission response rate, it's still 19%. I'll just articulate these because I don't have a pointer here. So still relatively, small complete remission response rate. But I think what really troubled us was to see what the long-term toxicities were from this trial. In fact, half the patients had reduced blood counts, we call those cytopenias, that were long-lasting. This affected, both white blood count, it affected, the red blood count as well as platelet count. But what really troubled us was to see the secondary malignancies, because this drug is considered an alkylator drug, and it has that sort of profile. Some of these malignancies you can attribute to people being older and being susceptible to secondary malignancies, and really important to keep that always in mind because age is always a variable for acquiring cancers. But what we were troubled by was the fact that we saw 3 out of the 69 patients have myelodysplasia, which is a precursor to acute leukemia. In fact, 2 of the patients went on and had acute leukemia. So I think whenever you have a discussion about which therapy, it is important to keep in mind not only the short-term, side effects, but also these long-term ones, and recognize that. And that's why with all our patients, we have these discussions when we're considering upfront therapy. Now, another, big question that emerged from our workshop is, do we put people on BTK inhibitors indefinitely, just like we do with, statins? Any of you on statins? I'm sure I am. I think most of us are. Cholesterol medicines, high blood pressure medicines, diabetes medicines, those are chronic therapies. And I always tell people, just because you're on a BTK inhibitor every day, it's not a bad thing. I mean, as long as it's controlling your disease and making you feel good. But there's also, a fair amount of interest these days in what we call fixed duration therapy. Not just, continuing on therapy indefinitely, but maybe fixing it for a definitive period of time. And so my colleague and good friend, Jorge Castillo, led what I thought was the first effort to be able to come up with a fixed duration therapy. And I really want to give Jorge a lot of credit, along with Shana Sorosiak in our group, for helping to navigate this trial, which combined ibrutinib with venetoclax. Venetoclax is a BCL2 inhibitor. Jeff already told you that is a very important target as well in Waldenstrom's. And this trial was intended to give 2 years of therapy and then stop. Unfortunately, even though we got really good results with this trial, we had an unexpected finding, and that is that patients got ventricular arrhythmias. These are bad arrhythmias. Atrial arrhythmias are when your heart skips and you have an irregular rhythm, but ventricular arrhythmias can be fatal. And in fact, we did have fatalities in this study. And so what I'm getting at here is that it's because of a clinical trial dedicated to Waldenstrom's that we learned this. When they did the same combination in CLL and mantle cell lymphoma, they didn't report any signals. But in Waldenstrom's, we saw a signal. And that's why we need to have dedicated trials for this disease, because sometimes the outcomes are very different than what other disease groups are telling us. And so as a result, this study was terminated. I think we learned a lot of valuable lessons, but the one thing we did learn is this was not a good combination for patients with Waldenstrom's. But I want to say now there has been a lot of interest in other combinations, and our friend Neil Berenstein in Canada leads a consortium in Canada that is looking at bendamustine with rituximab and another BTK inhibitor called acalabrutinib. Their trial is called the BRAHM study, so they put together all the acronyms. Oncologists love doing acronyms, by the way. You probably have already figured that out. But this has been an interesting study because they're giving 1 year of acalabrutinib with 6 cycles of bendamustine and rituxan. And we're actually very excited about their results. Neil did update his data at the 12th International Workshop in Prague. He has his data is now accepted for an update for a meeting in Switzerland, which will occur. Our big lymphoma meeting is in Switzerland in 2 weeks. But I want to tell you that major response rate was 95%. But even more importantly, what we saw from his data that excited us was the CR rate is still around 18%, but he was now reporting that in up to 23% of patients, they couldn't find evidence of Waldenstrom disease. Now, you might say, well, Treon, how do these numbers balance? I mean, you've got a higher MRD rate than a complete remission response rate. And that's true. There are some lag. Sometimes, disease detection, may be ahead of the curve from using traditional definitions of complete response, which depend on finding still residual IgM in circulation. And so this is one of the things that we need to, iron out in our assessment of responses. But I still want to tell you, the fact that almost a quarter of patients with this combination, it was hard to find evidence of disease, really bodes well for this combination. And for this reason, also, our colleagues in China have been experimenting with zanubrutinib along with bendamustine and rituximab. Shuhui Yi, who I think represents really the emerging light in Waldenstrom's in China, led this trial. He's based in Tianjin in China. And this was, again, in treatment-naive patients. They gave 1 year of zanubrutinib with 6 cycles of bendamustine rituxan. Again, very high response rates. 95% of the patients had a major response. But in the patients they looked for minimal residual disease, 50% were negative. And again, using technology to try to find the disease, they couldn't find it in about half the patients that got this combination. And this is one of the reasons why I think these kind of combinations are going to be very exciting going forward. Now, how do we manage high-risk disease? This is also a very important topic in our field, because there are patients who tend to do worse with our traditional therapies than others. And we wanted to help define what this population of individuals is. And at the 12th International Workshop, there was a report from Consensus Panel 3 that focused on identifying and managing patients with high-risk disease. And this was led by Prashant Kapoor from the Mayo, along with M.J. Kirsten from the University of Amsterdam. And a lot of this data in terms of high risk centers on this mutation known as TP53. If you're not familiar with this, gene, this gene actually codes for a protein, and some people call this the guardian of the genome. It's when a mutation occurs, TP53 goes in and sort of cleans up, that part of the DNA and fixes it. Elephants, which tend to have low risk of cancer, by the way, carry 40 times the amount of TP53 that humans do. And so this is just sort of a surrogate to show you why TP53 is very important. On the other hand, there are families that have what they call Leigh-Fraumeni syndrome, where you're actually missing TP53 that get a lot of different cancers. And so this is why, TP53 is very important. And the first actual investigators that recognized this in Waldenstrom's at the time was Xavier Leloup, who was one of my postdocs, along with his postdoc, Stephanie Poulain. And very early on, they published this paper, which appeared in Clinical Cancer Research, which showed that if you had a TP53 mutation or alteration, your survival was diminished. The time that you progressed after getting therapy was diminished. And so this really highlighted the importance of keeping TP53 in mind. The field really didn't adopt the TP53 testing for a while until other studies had come along. But I want to report on this now because at the 12th International Workshop, we learned two very important lessons. And that is that if you carry TP53, it's more likely to be in the setting of having been previously diagnosed and previously treated, I should say. And when you look at the patients that had the TP53 mutations, many of them had been exposed to alkylator drugs like cyclophosphamide and bendamustine. And in fact, almost a quarter of the patients also were exposed to nucleoside analogs like fludarabine or cladribine, other drugs that we don't commonly use today, but this was a study that took into account patients that had been previously treated as well as were treatment naive. So what did we learn from that study? And that is that if you had a TP53 mutation, you did actually better with zanubrutinib. This was one of those markers that actually patients did much better with zanubrutinib in terms of overall response and even length of response. But I want to show you now some updated data from this trial, and to highlight to you that even with zanubrutinib being such a very important and active drug, the TP53 mutated population shown in red there still seems to have a shorter progression-free survival, time that the disease is kept unchecked, relative to the other genomic groups. And when I talked about the MYD88 wild-type patients, and I said that they respond very nicely to zanubrutinib, look at also their progression-free survival median. It's about 39 months compared to those with the MYD88 mutation where it's 75 months. And so this identifies for us two very high-risk populations that I think we need to, as a field, continue to work and innovate on with drug therapy. So I'm going to actually skip over this treatment algorithm because I really want to move on and show you where a lot of the excitement is these days. And that is that if somebody progresses on ibrutinib or zanubrutinib, how are we going to treat them? And I think a new front has been opened with even more innovative BTK inhibitors. Leah Palumba, unfortunately, was sick and couldn't travel to be here with us this weekend. But she led this very nice study out of the Bruin study that specialized in Waldenstrom's patients, in relapsed refractory Waldenstrom patients, using this drug pirtobrutinib. Now, pirtobrutinib also binds to BTK, like ibrutinib and zanubrutinib does, but it's called a non-covalent inhibitor, and it binds to a different site than zanubrutinib or ibrutinib or even acalabrutinib bind to. Because of its ability to bind to these alternate sites, the drug is active in people that have mutations that otherwise would lead to loss of binding for those other covalent BTK inhibitors. What we've learned from this trial, and we also participated at our center, is that about 80% of patients respond. In fact, 77% response rate was seen in the patients that even saw a prior BTK inhibitor like ibrutinib or zanubrutinib. And the average benefit for these patients was about 22 months. So it shows you that even if you have, done very well on a covalent BTK inhibitor, you now have the opportunity to continue to have BTK inhibition using a drug like pirtobrutinib. So I think this is very exciting because it shows that we can continue to hit BTK very hard and make progress. Now, my colleague and friend Jorge Castillo also has been looking at the BCL2 inhibitor venetoclax. And this was a trial that we ran together, and this was a very exciting trial because it opened up the door for BCL2 inhibition, as Jeff told you earlier. And in this setting, over 80% of the patients still got a major response, even after seeing prior therapy. And in fact, when you looked at people that got a BTK inhibitor before, it was a little bit less, but still most of the patients responded. So it showed us that you could do actually quite well with venetoclax, and there was opportunity to get real good durable therapy. In fact, the average time to remain without progression was 30 months, so almost 3 years' time. And patients were only treated for 2 years on this protocol. So I am excited about venetoclax's entry. And so Jorge probably talked to you about the new generation of progress, combining some of these drugs, pirtobrutinib and venetoclax. He's going to have a very nice update at this year's American Society of Hematology. But I think this is really where we're going in terms of new combinations to treat people once they've progressed on zanubrutinib or ibrutinib. Because with this trial, even the snapshot that we got in Prague, 87% of the patients had a major response, 56% had what we call a VGPR, very deep response, and patients continue to do well on this trial. So we'll look forward to Jorge updating us at this year's American Society of Hematology meeting, but I do think this is what we're seeing on the future. Now, let me tell you about another very exciting drug, and this is a degrader. Now, we're going to switch because I want you to understand the difference between a BTK inhibitor and a BTK degrader. BTK is an enzyme, in essence. It can catalyze activity and make other proteins get activated and turn on all these signaling controls. But degraders actually go in, bind to the target, and chew it up. They destroy it. And what's exciting is that you can leverage this technology for BTK. Now, another good friend and colleague, John Seymour from Australia, reported on this new drug at the 12th International Workshop. We're also participants in this trial as well. And this drug is known as BGB-16673. This is also a product of B1 that is manufacturing zanubrutinib. And what got me very excited was when you see, in fact, in even these early Waldenstrom results, almost 75% of the patients had a major response. Most of these patients actually had seen a previous BTK inhibitor. This is really, very exciting. And in fact, when you look at all the different mutation categories there, BTK, MYD88, CXCR4, TP53, now you know about all of them, it made no difference. Patients responded despite these mutations. And in fact, when you look specifically at the mutations that normally would lead to resistance against either ibrutinib, zanubrutinib, or even pirtobrutinib, those mutations really didn't make a difference. You still saw very deep responses with this degrader. And so I do really see this as, eventually getting FDA approved. Right now it's investigational, it continues to be studied, but the results have been great, and even the safety profile on this looks extremely favorable. Another very important drug also out of B1 is sonrotoclax. And this also goes by the BGB acronym 11417. I can't keep track of these numbers personally, which is why I always put the parentheses there. But I want to tell you, this is also a drug which has the potential to replace venetoclax. It's a BCL2 inhibitor. With venetoclax, you need to be very careful in terms of ramping up the dose. Sometimes it can trigger something called tumor lysis syndrome, which means it works so good, the tumor cells die very quickly. When they die quickly, they can cause havoc. Sonrotoclax seems to have a slightly better profile in terms of ramping up. And its safety so far looks very favorable. So from Spain, we heard from Ramon Garcia Sanz in Prague, who presented the early results. These are phase 1 results of sonrotoclax in relapsed refractory Waldenstrom patients. And again, many of these patients heavily pretreated, but we saw wonderful activity. In fact, the overall response rate was almost 80% for this drug in relapsed refractory Waldenstroms. So this is also, I think, another drug which is destined to also see approval. At this point, it remains investigational, and patients are accruing on this study. Now, I want to tell you that when the debate is over BTK inhibitor versus BCL2 inhibitor, we should get excited about BCL2 inhibition. And this was a study out of our group, and I'll just summarize it rather than going through all the different points here. And that is that if you're assessing response by IgM, all the drugs look great, right? If you're seeing a reduction in IgM, you're going to check off and say they all look the same, right? Not really, because when you start looking for MYD88 as a marker for what's left in the bone marrow that's really Waldenstrom disease, BCL2 inhibitors seem to come out front. In fact, you see a lot more destruction and elimination of tumor cells with BCL2 inhibitors. And that's why we're very excited about the development of these BCL2 inhibitors, either alone or in combination, particularly with drugs that won't, give that ventricular arrhythmia signal that we saw with ibrutinib. Now, I want to, end up here by talking about the future, because I think it's always about the future, right? And the future is very bright. I want to leave you with that impression today. The future is very bright because we've learned so much about the basic biology of this disease that, shame on us if we don't find a cure, because that's how much knowledge we now have. And so let me tell you about the future. And a lot of this is going to revolve around having good medicinal chemists continue to work on Waldenstroms. And at Dana-Farber, we've assembled really a dream team. It was initially led by Nathaniel Gray, who's now back in his home state of California at Stanford, but continues to be shepherded by Sarah Berlagi, John Hatcher, who's actually in our laboratory, and Gino Wang. Those others are based at Harvard. Now, there are other targets. I showed you this signaling diagram earlier, and I got you all interested in HCK. I said this was kind of a villain that's turned on by MYD88. But it's really a therapeutic opportunity as well, because if you look at that signaling diagram, what do you see? You see that HCK turns on BTK. You see that HCK turns on this other molecule called ERK. ERK actually is a bad player because it's the one that releases all the inflammatory proteins that help protect it in the bone marrow microenvironment. But it also turns on this other molecule called SEEK, and SEEK itself also promotes the growth and survival of Waldenstroms. So instead of going after BTK, which is further downstream, let's bring up our therapies and target some of these upstream bad players. And HCK represents one such bad player. And the IWMF, along with the Leukemia Lymphoma Society and the NIH, provided us funding to be able to go after this target. So initially, we developed this drug called KIN8194. It's an enzyme inhibitor, kinase inhibitor, much like a BTK inhibitor that we talked about earlier. And we were very excited about this drug because it did something that we don't normally see when we do these kind of drug studies. When we evaluate drugs in mouse models, normally what you see is that the growth of the tumors that we put in are slowed down. Here we were actually seeing cures of many of these mice. And so this really did tell us that if we could move our targeting of therapy more upstream, closer to MYD88, we have the opportunity really to silence a lot of those survival pathways that a BTK inhibitor would not do alone. And now these days, it's all about degraders. And so John Hatcher is one of the world's top experts in making degraders. And so he has been actually trying to make degraders that actually go after HCK, but also include BTK. And I want to tell you about one of these projects which the IWMF helps support along with the LLS, and it has led to the discovery of this molecule called DFCI 00206. Again, it's all about numbers and acronyms, right? And so let me tell you about this drug. It actually does a beautiful job of destroying both BTK and HCK. And again, I don't have a pointer here, but that red box that you see there shows what happens with degradation. You don't see any protein showing up on these Western blots, but what you do see is killing. And that's why we're very excited about this drug, because when we put these tumors in mice that have the MYD88 mutation, these lymphomas, actually we can destroy them by using these degraders. And you give the drug actually orally, much like we would hope to do with a pill, where the drug actually is ingested, it goes through the circulation, and it actually gets into the tumor. And once again, look up there and you see both HCK and BTK in the tumors themselves. The proteins are destroyed. They're decimated. And so as a result, as we look to the future, I think the ability for us to drug HCK, even in combination with BTK, looks very attainable. And so we want to now move these discoveries to human trials, and that's what gets us all excited. I also want to tell you about MYD88 itself. If this is public enemy number one, right, mutated MYD88, let's go for it, right? And this is why I want to first extend my appreciation to the IWMF, because it's the first agency that has funded an effort to drug MYD88 itself, the mutated form of MYD88. Now, this is not an enzyme. MYD88 is not an enzyme. So really the most tenable way to go after it and drug it is by destroying it. And if you can actually destroy the mutated form, because there's also, we need the wild type, the regular one, to help us fight off infections. But if we can destroy the mutated form, then we've really got a very targeted therapy. And I'm actually excited about what we've learned. We put together also a dream team around this. These are protein chemists based at Harvard, Jared Mardo, along with Serrano de Paganon, and they run this magnificent laboratory which actually goes through thousands and thousands of compounds, and they try to find which ones actually bind to MYD88. And as a result, what I'm showing you here is all the compounds they initially discovered that actually targeted the wild type, the normal form of MYD88. It's been a little bit harder for us to manufacture the mutated form in the batches that we need. We finally have been able to do that. But I want to show you, even going after the wild type, look at all the compounds that they have already identified for us. And so this is why I'm very excited about moving this field forward, because getting drugs that can specifically go after the MYD88 mutated form, I think will open up many, many doors for us, and I'm truly excited by these efforts. What other drugs can we get excited about? Well, cellular therapies. I want you to know that this is a front which we need to open for our field. We've seen tremendous success in myeloma. We've seen tremendous success in lymphoma. We're now going to be bringing this also to Waldenstrom's. And in fact, this October, we hope to have the first ever meeting devoted to cellular therapies, bringing an advisory group to help us focus on which drugs we need to develop. So on both sides of the Atlantic, Josephine Vos in Europe, Gottfried von Kudel here in the US, he's based in our Harvard consortium, are developing these bispecific antibodies. What do these drugs actually do? They actually bind to the tumor cell, and they bind to the immune cells. They bring them together to be destroyed. And so this represents a phenomenal opportunity for us, because in myeloma, they're seeing 80% complete remissions, maybe even 60% MRD negative disease. And that's a disease that's actually even more challenging than Waldenstrom. So I do really believe in the future for these bispecifics. And you'll be hearing more about them. There's many people that are part of the WMNet that have already introduced new concepts for developing these drugs. But trust me, this is going to be a very exciting new horizon for us, and we'll reap many rewards going forward. You've heard of CAR T cells, right? Yes. Do we have any active CAR T trials right now in Waldenstrom's? No. And this is a challenge for us because we know CAR Ts work. In fact, Mazar Shadman did a very small trial with CD20-directed CAR Ts. These were heavily pretreated patients. In fact, on average, some of his patients had seen 8 prior therapies, on average. In fact, one patient saw 12 prior therapies. And yet he had an incredible response rate, 67% response rate associated with CAR Ts, even in these very heavily pretreated patients. Unfortunately, this also got a breakthrough and was supposed to go forward, but the company was no longer able to fund, this trial, and this trial had to be closed. Now, there's a lot of good CAR Ts that are out there that need to come to Waldenstrom's. But the issue that I find as we talk to all these companies is, well, it's a small disease. You're doing well with BTK inhibitors and BCL2 inhibitors. We'll call you when we think it's time. Well, it's time now. And this is where I think the foundation can play a very active role, along with all of you here, really in calling attention to the fact that these drugs need to be investigated now. And with this also comes the responsibility of being able to get patients on these trials. And as I say, the challenge today for us is not only to bring some of these innovative new therapies to Waldenstrom's, but also to be active participants in these trials, because this is the way that progress gets made. So I want to tell you, in our group, we're already thinking ahead, because as we see, one of the holdbacks of these cellular therapies is that the immune microenvironment in Waldenstrom's is very suppressed. We're not seeing the activity that the immune system should be mounting, in these in our patients. And so in our group, and also funded by the IWMF, is for us to understand why that immune system has been dampened. And I got to tell you, when we come back next year and we talk about this progress, I will be able to tell you what Waldenstrom cells are secreting and what they're doing to these myeloid-derived suppressor cells that are then turning around and suppressing the immune system. And in fact, we have some very exciting data around this. And this phenomenon is called T-cell exhaustion. At the end of the day, what you're doing is you're taking all your T cells, your immune cells that are supposed to recognize and go after the Waldenstrom cells, and we're tiring them out because of these proteins that are being made and released by the Waldenstrom cells, and they're duping these myeloid cells to be part of that, exhaustion phenomenon. This we need to reverse, because any one of those therapies that I just showed you is very much dependent on us being able to turn around and get the immune system primed up again. And so I'm very excited by some of the initial results we've seen. Steve Ansel and his group have also been doing a wonderful job trying to understand myeloid-derived suppressor cells in their work at Mayo. And so I do think when next year comes around, we'll have a lot of exciting data to share with you. Now, I'm going to end here by telling you that the future is bright, and for one other reason. And I think Zach Hunter should be given a lot of credit, because what he's taught us through his multi-omics studies and pseudotime-derived tumor evolution studies, and you can read more about that from, in his upcoming paper, is that Waldenstrom's, as we know it, probably is 3 molecularly distinct diseases. There's this early group, there's 2 other later groups, one called BCL-like and one called plasma cell-like. And I just want to go to the next slide here before I conclude by telling you that these distinct groups being identified will allow us to make more targeted therapies for each one of these subgroups. The early Waldenstrom groups has its own, molecular signature. But as you look at the later groups where the early Waldenstroms evolves, one looks more like a B cell and one looks more like a plasma cell. And each one of these will offer us new targets going forward. And I am really excited to also see how we're going to use this in being able to define even how current therapy should be managed. If you have a B cell like Waldenstrom's that needs treatment, you get managed differently. These are patients, by the way, that have a lot of CXCR4 mutations. If you're plasma cell-like, you don't see as much CXCR4, but you see more chromosome 6 deletions, which occur in about half of all Waldenstrom patients. And there are certain genes that get lost in these patients. But I will tell you, we will have to define as we go forward our disease and our progress based on these molecular classification. Our next meeting will be held in Palm Springs, California. I expect it to be full of lots of excitement based on all the progress being made. And I am happy to report that Steve Ansel, Prashant Kapoor, and Ranjan Advani from Stanford will be chairing our next meeting. And with that, I want to stop and again thank the IWMF for really being at the center of all this progress that's being made, and to encourage each and every one of you to support the IWMF in its mission and to participate whenever possible on clinical trials, because that's the only way we're going to be able to find and make a cure. And with that, I'll stop, and thank you so much for your kind invitation to be here. Should we take some questions, or
Do you want to ask a couple questions?
if anyone has questions here?
Dr. Treon would love to have a couple questions but we're only going to have a couple, guys, because he would stay here all day with you. So.
Since most Waldenstrom's patients have this mutation, this bad actor MYD88, is there an understanding of what caused this mutation in the first place?
No, it's a very good question. And we're very interested in this, in the predisposition. MYD88 is part of a pathway that is supposed to help us. Whenever you have an infection, your B cells are part of the response to infection. And in fact, MYD88 is among the first alarm system. So one potential explanation is that somebody gets an infection, and unfortunately, during the process of those B cells expanding and expanding and expanding to go after the infection, that because you're triggering this pathway, the MYD88 protein, there's a genetic mutation that occurs because you're stimulating now cells to divide, and every time cells divide, you can see mutations set in, and particularly maybe in people that have chronic infections where you're seeing this ongoing firing. Now, at odds maybe with this is the fact that you also see familial Waldenstrom's, and in fact, maybe up to 20% of patients have a family member with Waldenstrom's or another type of lymphoma, and it might be that they particularly are vulnerable to this sort of cascade of, chronic infections triggering, the MYD88 signaling. And so this is something that we're looking at. We, in fact, have one gene that we're very suspicious of, and we're working on that. But, in our group, we have almost 250 families with familial Waldenstrom's, and so it's a great resource for us to be able to interrogate and find out why we have familial WM.
Dr. Treon, this is a different kind of question. I don't want to be overly political. You celebrated FDA approval processes and NIH and all that. There's this little university you're associated with that may not have the best favor at a certain address on Pennsylvania Avenue. Whatever is happening in DC, is it affecting anything that you all are doing? Do you see threats to processes, basic research, approval processes, et cetera? And if so, what do we do about it?
Yeah, no, I think the foundation particularly has a role in this because we're all being affected by, and it's actually beyond just the funding elements of this, it's also the talent. You know, a lot of the talent that we actually get in our laboratories are from overseas. And China in particular has really contributed a lot of talent to our laboratory. And so I think this is also part of, you know, where, you know, there's a lot of concern in terms of being able to bring these individuals to work in our laboratories. And for Waldenstrom's being a rare disease, you got to understand you really have to bring this talent because there's really, you know, a finite number of people out there that are excited about our disease. Now, I want to also, in that same breath, say that in Prague, again, thanks to the IWMF, we had 32 young investigators that came, and they were funded through an award that the IWMF made possible. And we were able to bring those young people, and they had some brilliant projects that they reported on, and they're interested in a career. So we have Waldenstrom, we have, you know, people interested in Waldenstroms all over the globe, and being able to collaborate and work together is quite important. From a funding perspective, it's more crucial than ever now to recognize how the foundation can make a difference, because I fully expect there'll be far less money coming out of the NIH. It's already been a trickle for Waldenstroms all along, so don't think, even though I put the logo up there, you know, it's been a trickle. It's been mainly the IWMF and the LLS and philanthropy that has made all this possible. And I continue to encourage you to support the IWMF and make a difference, because this is the only way we're going to be able to get to this cure. And we will. I really said, you know, shame on us if we don't find a cure, because look at how much progress we've made. And that's why I'm more optimistic than ever. I've been doing the IWMF meetings, by the way, the forums, for almost 30 years now. 30 years, right? 30 years. And so,
I think there's just one last question.
yes,
because we're-
yes.
Hear you.
Make up your mind.
Oh, no. Okay.
I had a question about TP53.
Yes, please.
So you were saying that Xananarup, I can't say it, was the best for TP53 mutation?
Yeah, so in that trial, the Aspen trial, the patients that, because it was a randomized trial, patients either got ibrutinib or zanubrutinib. The patients that got zanubrutinib fared better. They had better, deeper responses, and they actually had longer lasting remissions on zanubrutinib. Yeah. But then I showed you the next slide that showed that even though they did better on zanubrutinib, this is still a very high-risk population, because the time that their remission lasted was shorter compared to the other genomic subgroups.
Are there other trials going on?
Yeah, there are no other trials at this point that are focused on TP53, but this is one of the you know, as we also mentioned, and I was at the ECWM meeting last week in Paris, this is going to be one of our focus to try to develop specific trials for this population. I am encouraged, by the way, by Shana Sorosiak's trial. I didn't have a chance to talk about that, loncastuximab. It's an antibody that's conjugated to a drug toxin that kills Waldenstrom cells, and it doesn't really care what your underlying genetic mutation is. And she's actually, you know, surreptitiously, her trial is actually overweighted in TP53 mutated patients, and they're actually doing quite well. So I do think we will have an avenue forward there, but being able to have dedicated trials for high-risk disease is a huge priority for us.
Thanks.
Want a last question? Or?
It's up to you guys. Okay, real quick. So wild type. Do you really believe the 3%, 3 to 5% are truly wild type, or is it could possibly be a problem with how the bone marrow was, or it wasn't missed, all that? Because it seems like they're being included in trials and doing somewhat well with some of the drugs, but what is it really?
I love this question more than you know, because it really comes down. So, I mean, just to clarify, how we test for MYD88 is very important to whether we find it. Patients who have very low volume of disease in their bone marrow can often be missed. The mutation can be there. But if you want to be really stringent, you take out the Waldenstrom cells and then you test. And that's very hard for clinical laboratories to do. So the best technology is PCR technology, because your likelihood of picking it up is much better than if you're using next generation sequencing NGS, where you can miss up to a third of patients. And yes, it has troubled some of the trials where patients who are considered to be MYD88 wild type probably had the mutation, and that's why those, you see this sort of exaggerated response rate. But it is important for us to identify who is truly MYD88 mutated versus wild type, because you did see they also are a high-risk population. We want to make sure we correctly identify them.